Have you ever wondered how scientists actually manage to solve microscopic mysteries? Think about it. They're tasked with catching and identifying organisms that are literally invisible to the naked eye. In today's Explainer, we're diving straight into exactly how clinical microbiologists pull this off. Spoiler alert, it mostly comes down to manipulating bacterial diets. We're going to explore the absolutely fascinating world of culture media, which is essentially the process of creating highly specific menus to force microscopic culprits out of hiding. Let's get right into it. So how do you trap an enemy you can't even see? It's a pretty incredible puzzle, right? You obviously can't just set up a physical trap. Instead, microbiologists act as molecular detectives. They use specialized food, meticulously crafted chemical environments to coax, trick, or literally force these invisible culprits to reveal themselves, multiply, and confess their identities. To pull this off, we need the foundational tool of the laboratory, the culture medium. Now, by definition, a culture medium is just a prepared nutrient material used to support the growth, isolation, and identification of microorganisms. It can be liquid, semi-solid, or solid, but to really capture a suspect, you usually need a solid surface. Enter agar. Agar is a solidifying agent, and let me tell you, it's vastly superior to older methods like gelatin. Why? Well, because agar won't melt at ordinary laboratory incubation temperatures. And brilliantly, it's generally not eaten or degraded by the bacteria themselves. It just provides a perfect, stable stage for those microbial colonies to form. Section 1. Welcome to the microbe menu. Depending on exactly what the lab needs to test, these microbial diets can take a few different physical forms. First up, you have liquid media, which are often just called broths. Then you have solid media, which use that amazing agar we just talked about. And finally, you have semi-solid media, which use a reduced agar concentration. These semi-solid gels are actually incredible because they're primarily used for motility testing. It's like giving bacteria just enough space to swim around so we can visibly see if they're mobile or not. Now, if you're feeding bacteria that aren't particularly picky, you just use basal or simple media. Think of these as the standard, everyday meals of the microbial world. They're used for routine cultivation, basic biochemical testing, and really just maintaining easy-to-grow, non-fastidious organisms. Common examples you'll see around the lab include nutrient agar, nutrient broth, and peptone water. Section 2. Feeding the picky eaters. Now, what's really interesting about this slide is that it clears up a massive point of confusion in the lab, the difference between enriched medium and enrichment medium. I know they sound absolutely identical, but they do completely different jobs. An enriched medium is typically solid. It's a basal medium supplemented with extra rich nutrients, like blood, specifically to support fastidious organisms. Those are the highly picky eaters. An enrichment medium, on the flip side, is usually a liquid broth. It's designed to selectively enhance the growth of one specific pathogen out of a mixed specimen. It's literally like trying to find a needle in a haystack and giving that needle a megaphone. Let's look at one of the most famous enriched media out there, chocolate agar. First things first, we have to get this out of the way. It does not contain any chocolate. Zero. It gets its name purely from its brown color. And that color actually comes from heated blood. You see, highly fastidious organisms like Haemophilus and Neisseria species need very specific nutrients. By heating the blood, the red blood cells literally burst open and release two absolutely crucial high-yield growth factors, the X factor, which is Haemin, and the V factor, which is NAD. Without this precise culinary preparation, these picky pathogens just wouldn't grow at all. Another critical enriched diet is standard blood agar. But here's the cool part. Blood agar doubles as a lie detector. It reveals exactly how a bacterial suspect attacks red blood cells, which is a process called hemolysis. Take a look at the breakdown. Notice how alpha hemolysis is just a partial breakdown? It leaves this distinct greenish discoloration around the colony. Then you have beta hemolysis, which is complete destruction, leaving a stark, completely clear zone around the colony. And gamma hemolysis means no breakdown at all, so the red medium stays perfectly intact. This visual clue is an immediate dead giveaway for our laboratory detectives. Section 3. The interrogation room. Welcome to the interrogation room. This is where we use brilliant chemical traps. First, we have selective media. These act like a highly strict VIP bouncer. They contain specific substances that actively inhibit certain organisms while permitting the exact ones we want to see to grow. Then we have differential media. These act more like a chemical lie detector. They allow different organisms to grow side by side, but visually distinguish them based on observable biochemical reactions, which is almost always a very obvious color change.
And the undisputed king of this interrogation room is McConkey agar. It's one of the most critical tools in clinical microbiology because it performs two interrogation jobs at the exact same time. It's both selective and differential. The bouncer aspect inhibits gram-positive bacteria, allowing gram-negative enteric, or intestinal, bacteria to grow. Then the lie detector aspect differentiates those gram-negative survivors based on whether or not they can ferment a specific sugar called lactose.
And this brilliantly illustrates the true power of differential media. If the bacteria ferment lactose, they produce acid, and the indicator in the McConkey agar turns the colonies a bright, unmistakable pink or red. This points the detective right towards suspects like E. coli or Klebsiella. But if they cannot ferment lactose, the colonies remain totally colorless or pale. And that points toward entirely different, often much more dangerous suspects like Salmonella or Shigella. Just by looking at a color change, you instantly narrow down your suspect list. It's incredible.
Let's look at another ingenious dual-purpose trap, mannitol salt agar. Here, the selective bouncer is a really high salt concentration. Most bacteria absolutely hate this, but staphylococci, they thrive in it. Once the staph is isolated, the differential lie detector kicks in. Can the bacteria ferment the sugar mannitol? If it can, it produces acid, and the agar right around the colony turns a vivid, bright yellow. This is a classic, rapid way to flag a potential Staphylococcus aureus infection, which is absolutely vital for catching common and sometimes really severe skin infections in patients.
Now, sometimes a lab faces a highly specific nightmare scenario. Enter CLED agar, which stands for cysteine lactose electrolyte deficient agar. It's absolutely crucial for urine cultures. Now, it differentiates lactose fermenters beautifully, but the real secret weapon here is at the electrolyte deficient part. Certain highly motile bacteria, specifically Proteus species, love to literally swarm rapidly across an entire petri dish, covering up all the other evidence. By removing electrolytes from the agar, CLED chemically paralyzes the Proteus, stopping the swarm dead in its tracks. A real game changer. Section 4. Highly specialized diets. Microbiologists constantly have to adapt the chemical environment to solve highly unique challenges. If you're hunting for Mycobacterium tuberculosis, you use Lowenstein-Jensen medium, which is an egg-based solid that supports its painfully slow growth. What about fungi like yeasts and molds? You use Saborodextrose agar, which is highly acidic to kill off any competing bacteria. For anaerobic organisms that literally die in the presence of oxygen, you use thioglycolate broth, a reducing medium that creates a safe, low-oxygen environment. And if you just need to transport a specimen across town without the bacteria multiplying and ruining the ratios, you use transport media like Amy's or Carrie Blair. It's all about picking the perfect environment for the job. Section 5. Reading the clues. After the incubation period is over, the scientist examines the physical mugshot of the bacterial colony. We call this colony morphology. They meticulously document the size, the shape, the specific edges or margins, and the elevation. They note the color, how opaque it is, and its physical consistency, like is it dry and brittle or is it wet and mucoid? Every single physical trait is a vital clue in the ongoing investigation. But listen closely, because this brings us to the absolute golden rule of the clinical laboratory. Never, ever jump to conclusions based purely on color. Seeing a pink colony on McConkey agar does not automatically mean you have E. coli. It simply means you have a lactose-fermenting gram-negative bacillus. Sure, it could be E. coli, but it could also just as easily be Klebsiella. Guessing the exact species strictly from the agar plate is considered a massive professional error. You just can't do it. So, the crucial point is that seeing the growth is only step one. The proper identification protocol requires strict discipline. First, you observe the colony morphology and the reaction on the differential medium. Second, you perform a gram stain to physically confirm the cellular structure under a microscope. And third, you execute specific targeted biochemical tests to finalize the exact species-level report. Only then, after all that, can you officially convict the microscopic suspect. It's an incredibly meticulous process, isn't it? And it leaves us with a truly fascinating thought to wrap up this explainer. If clinical microbiologists require such incredibly specific, wildly diverse diets just to see the microbes we already know about, how many invisible organisms, hidden pathogens, or perhaps even life-saving bacteria are still out there, entirely undiscovered, simply waiting for us to figure out what they eat? Think about that. Thank you for exploring the micro menu with me today. Keep asking questions and keep looking closer.